Hi guys. So we're going to talk about basically the last system here, and it's the reproductive system. And we're going to start with male anatomy and physiology. We will be talking about female anatomy and physiology in the second part of this. So let's discuss a little bit or watch this video on why this subject even matters.
Did you know that the United States has the highest rate of sexually transmitted infections among developed countries? Sexually transmitted infections, or STIs, are the most common disorders of the reproductive system. Since over 12 million Americans contract STIs each year, it is highly likely that you will encounter some of the most prevalent STIs in your patients, including gonorrhea, chlamydia, the human papillomavirus, and herpes. As a healthcare professional, it is very important to educate your patients on their reproductive health. In order to do this effectively, you will need to understand the underlying infectious agents, modes of transmission, and symptoms of the most common STIs. For example, gonorrhea is caused by a bacterium that is spread by contact with the mucosal surfaces of the reproductive system. These bacteria invade the mucosa of the reproductive and urinary tracts. Since gonorrhea is caused by bacteria, it can generally be treated with antibiotics. However, if left untreated, gonorrhea can cause significant complications like infertility in women and inflammation of the entire male reproductive tract. Having a solid understanding of the anatomy and physiology of the reproductive system will enable you to recognize symptoms of reproductive disorders like STIs, give you the ability to treat your patients with STIs, and educate all of your patients on ways to maintain a healthy reproductive system.
So hopefully that kind of gives you a good idea why this section is important. Now, when we're talking about the reproductive system in general, we're talking about primary sex organs and basically what allows us to reproduce. And those primary sex organs, known as gonads, are different for male and women. In men, they're called testes, and in women, they're called ovaries. And they both produce what's known as gametes. And gametes are basically just sex cells. So I like to say how I always remembered this is sex is a game. It's not the most PC way of saying it, but that's how when I was going through, I remembered what gametes were. Now, gametes are basically sperm and ova. Sperm being the basically sex cells that are produced in males and ova being in females. Now, the sex hormones that help with this entire process that are produced are both known as androgen, which is the sex hormone in males, and estrogen and progesterone in females. So since we're going to start, that was kind of an overview of both. Now we're going to get more specific here in a second, but essentially there are accessory organs for each male and female. And those accessory organs that we're going to talk about in each case are the ducts, the glands, and basically the external genitalia. So now that we've kind of given a brief overview, let's go into male reproductive system, and we're going to start with talking about the testes. And so basically, the thing that produces sperm in men are the testes. Whoops. Oops. Oh, it's gone all over the place. There we go. And these can be found within the scrotum. And like I said, they produce sperm. And basically, sperm is delivered to the exterior through a system of ducts. And so if you look at this picture, I've kind of shown you basically the duct system that they start with. And so once it leaves the testes, it goes to the epididymis, you can see here, and then it's going to leave out of that and go up here to the ductus deferens, known as the vas deferens. It's the second one. From here, it'll go through here, and basically next it's going to go into the ejaculatory duct. And then finally, it's going to go out into the urethra. Now, this is the duct system that it goes through, and there are also accessory sex glands in here that basically help with the movement of sperm through this. So you can see up here, you have the seminal gland. You have here the prostate gland, the bulbourethral gland here. So those are the three main accessory sex glands that we're going to end up talking about here later in this lecture. And so let's talk about the scrotum. So this is a sac of skin and basically superficial fascia, right? It hangs outside the abdominal pelvic cavity. Let me change. It basically contains paired testes. Essentially what it is, the scrotum has this midline septum that divides it in half, and then you get each side has one testes within it. Because they're able to hang a little bit lower, they're 3 degrees Celsius lower than the core body temperature. And the lower temperature is necessary to help with sperm production. And so basically, the temperature is kept constant by 2 sets of muscles. The darto muscle is the first one, and this is composed of smooth muscle. And essentially, it helps to wrinkle the skin. And so what this does is it helps to pull the scrotum closer to the body. Now, the cremaster muscle is skeletal muscle. And this basically helps to elevate the testes. So these guys together will work to either make the scrotum shorter or more wrinkled. And if that happens, it'll pull it closer to the body, so the temperature will increase, or it'll become flaccid and loose to increase the heat loss when it's too warm, so it'll hang lower. So these muscles work together to allow the temperature of the testes to remain pretty much constant. So now let's look at the external part to kind of get an idea of the male perineum. And so this is the diamond-shaped region between the pubic symphysis. So this is the pubic symphysis, that's right here. The coccyx, which is back here, and the ischial tuberosities, which are here. So if you look, it's kind of that area that's right here, which this is the reason why we talk about bone structure as well in part 1, because then it'll help you to start getting some reference points when we talk about the anatomical structure in other parts of the body. And so in this area, this contains basically the root of the penis and anus, and it also is going to, this should be down a little bit farther so you can see, and it's the area where the scrotum is suspended. So that's if we're looking at that area just on the outside. Now, when we're talking about the testes in particular, right, this is what we find inside of that sac or the scrotum. Each testes is surrounded by some tunics. And so you have the tunica vaginalis, and basically this is the outer, and you can see this over here, right? The outer layer, and this is derived from the peritoneum, and you can see that right here. And so it's really kind of harder to see, but you can see it's pointed in there. And then you have the tunica albuginea, right? And this is the inner layer. And this forms basically the fibrous capsule of the testes. Now, there is a septa within the testes that will allow it to be divided into about 250 different lobules. And each one of these lobules contain 1 to 4 seminiferous tubules. And the seminiferous tubules are important because this is the actual site of sperm production. And if you remember earlier when I was talking about the midline septum, that's the midline septum. You can kind of see in there, right? It's also showing you right there. You can also see on here that you have basically the different muscles here, right? Shows one of those muscles there. If there's anything on there else that we've talked about yet. And here's the other muscle right here. That is talking about the scrotum and the skin. So, and that might actually help you there too when identifying what each muscle does. Now, let's kind of take a little bit of a look. We're going to, here in a few minutes, get closer into the testes so that you can see these lobules and the seminiferous tubules. But first, we're going to talk about what important cells are within the seminiferous tubules that help with the production of sperm itself. And you can see this whole picture here. This whole area right here is the seminiferous tubules, right? And so in the epithelial lining here, we're going to talk about first the separated spermatogenic cells. And these are the actual sperm forming cells. And so you can see those right here, right? The next one we're going to talk about, the sustainocytes, and you can think they help to sustain the cells there. So these are supportive cells. And so you can see those. Let me put this as 1 and 2. So this is 1 and this is 2 here. You can see them up there, right? Now surrounding those cells are 2 different types. There's myeloid cells and then there's the interstitial endocrine cells. So these myeloid cells actually surround each seminiferous tubule. So you'll see those right here, right? And you can see them located in this area right over here. And these are basically smooth muscle-like cells. Let me put smooth muscle-like cells, right? So basically, they are thought to help squeeze sperm and testicle fluid, or testicular fluid, out of the testes. Now the last one is here. These interstitial endocrine cells, which are also known as Leydig cells. And they're located in the soft tissue surrounding. And they're important because they help produce androgens, such as testosterone. And they secrete it basically into the interstitial fluid there. And here right now, we're just looking at where they're located. Here in a few minutes, when we actually get to the physiology part, we'll talk about what all these things are and how they're important. So when we're talking about making sperm and trying to make it so that it moves around, we're going to talk about what compartment it goes to. So essentially where it starts when we make them is the seminiferous tubules. And you can see that located right here. So all these little lines that you have up here, right, are the seminiferous tubules. And if you look here, there's the lobules. So you can see the seminiferous tubules located there, and there's several lobules there, right? And so basically what happens, it goes from there into the straight tubules. So you can see the straight tubules here in this location, right over here. Those very small things that are going to connect to this larger unit here, which is called the ret testes, which is where it goes next. From here, it's going to go into these other straight kind of lines here called the efferent ductiles. And then the last thing it's going to do is go through the epididymis. Now, the epididymis actually is made up of 3 parts. It's made up of a head, a body, and a tail. And so if you look at where the efferent ductils are, you'll notice that they come in contact with the head. So it's going to go from the head down to the body to the tail. Now the sperm are actually stored in the tail until ejaculation. Now they can be stored there for several months. And basically there are these little microvilli, or also known as stereocilia. that will absorb the testicular fluid. Basically, from there, they will pass the nutrients from that to the sperm. And so the sperm will enter non-motile. And they will pass slowly through this. It takes about 20 days. And they'll go from non-motile to being able to move. And so during the process of ejaculation, when the sperm moves from these, basically it's going to leave the epididymis and go into this area at the top called the ductus deferens, or vas deferens is how I remembered it. So let's go on now from the epididymis area to the ductus deferens or vas deferens. And we're going to talk a little bit because this is the duct system that we're going to get into. So the ductus deferens or vas deferens are smooth muscles. And within that, the walls that contain the smooth muscles, they can contract, which will propel the sperm from the epididymis to the urethra so that it can leave. And so if you look at this, we're going to start, if you look here, that part right there is what we were just looking at with the epididymis. So basically it's going to go from there to into that guy, which is the ductus deferens or vas deferens. Now the vas deferens will expand whenever you come up here. So do you see that thing right there? And that's what's known as an ampule. So this ampulla will kind of allow it to basically just open and get bigger a little bit there. Now from there, you're going to see it's going to go into this area right here, which is known as the ejaculatory duct. Now, this, if you notice, basically, get a different color here. There's this guy that's right here. So basically, the ejaculatory duct is the ampulla plus the seminal gland. And we're going to talk about what the seminal gland does here in a second. So they come together to make this ejaculatory duct. And then basically from there, they're going to go into this part right here, which is known as the urethra. Now I'm going to show you here in a bit that as the urethra goes down here to exit, that it is subdivided into several different parts. And we're going to get to that here in a second. And I'll show you a little bit. Now, there are some ways to essentially help in effective birth control, changing the duct system or cutting the duct system off. So there's what's known as a vasectomy, and this is when you're cutting or ligating the ductus deferens. And so if you cut this, it is essentially nearly 100% effective as long as it remains cut. They even have in other countries that have not been approved here, a polymer that they essentially can inject in this area, and it basically blocks the movement of sperm from here. And you can keep that until the point that you want to have children, and then they inject you with another solution that basically breaks down that polymer and allows you to have kids. It has not been approved in this country. I know they've used it in certain parts of the Middle East. I believe they've used it in India, but it hasn't been approved here for use yet. Now, when we're talking about blood supply, nutrients, and oxygen, and how everything is maintained in the testes, the arteries, the testicular arteries, basically arise from the abdominal aorta. And you'll remember this, that main area that comes out in the back. Now, the testicular veins actually rise from the papiniform venous plexus. These basically, this is surrounding each testicular artery, and it helps a lot. It helps to basically keep it cooler. It can absorb heat from the arteries, and that essentially will make sure that the testes remain cool. And so when we're talking about where these are located in, you can see it within the spermatic cord. So that is here, and you can see that it comes down, and this is what encloses the nerve fiber, the blood vessels, and the lymphatics that actually supply this guy. Now let's talk a little bit about the homeostatic imbalances that can happen to the testes. It's rare, but testicular cancer is the most common cancer in men from the ages of 35 to 50. Having mumps that leads to orchitis, that's an inflammation of the testes, could be a risk factor. Cryptorchitism is the most common risk factor on the descent. Signs of this are basically painless and solid masses in the testes. And basically, it can have a 90% cure rate by surgically removal of the testes and other radiation and chemical therapy. So testicular cancer doesn't necessarily mean a death sentence. It is completely treatable. So now let's actually look at the penis itself. So, and the overall organs. So when we look at this, the penis is the copulatory organ, right? And so when we're talking about the external genitalia, that essentially is talking about the scrotum and the penis itself. Now the penis consists of the root and the shaft and ends in what's known as the glans penis. Now the prepuce is also known as the foreskin, right? And this is a cuff of loose skin. So this covering here is what gets removed during a circumcision. And this is kind of interesting. It's pretty common in the US. About 60% of the male population in the US is circumcised. But if you look at the world, it's only about 15%. So no, not as common. Now, there is striking evidence out there that says that circumcision can actually help reduce by 60% the rate of HIV risks. And it can actually reduce other reproductive infections. Now, internally, the penis is made up of basically the spongy urethra and 3 cylindrical bodies of erectile tissue. So if you look here, the spongy urethra is right in here. And then when we're talking about the 3 cylindrical bodies, you can see down here what we're talking about, right? The 1, 2, and 3. They're kind of showing you the different, and we're going to look at what these guys are made up of, right? And so these are just spongy networks of connective tissue and smooth muscle. And they're going to have some vascular space in there. Now, when we're looking at them, one part is what's called the corpus spongiosum. And so if you look, that's the guy right here, and you can see it's on this outside, right outside that spongy urethra. And you can see it down here as well when we're looking. And so you see the urethra kind of in that circle right there. And then right there in that basically third circle, you can see that corpus spongiosum. And so this is the part that essentially expands to form the glands and the bulb. And you can see that down here. Now, the corpora cavernosa is right here. You can also see this in this section here and this section down here, right? In 1 and 2. And these are basically paired dorsal toward the back, erectile bodies. And so these are going to help with forming the erection. And basically what happens is erectile tissue fills with blood, causing the penis to enlarge and become very rigid. Now, if you look at this right here, the crus, or what's also known as a cura, this is the proximal end of the corpora cavernosa. I'm just going to put the CC, right? And this is what essentially anchors the penis to the pubic arch. So this kind of gives you an idea of the, what's on the outside, what's actually making the shaft of the penis, and how it's connected to the body. So now that we know the structure, let's focus a little bit on the different parts of it, specifically the urethra. And I had told you earlier when we talked about it going through here, we would get into the different parts. So the urethra conveys both urine and semen, of course, at different times. It has 3 different regions. You have the prostatic region here, and this surrounds the prostate. And the prostate's a gland that we're going to talk about here in a few minutes. The intermediate part, two, of the urethra. And this basically, as you can see, it is into your general diaphragm, or the triangular ligament muscle. So you can see right here that it's basically the area that is around here. And the last is the spongy urethra. And this runs through the penis. And it opens at the external urethra orifice. So you can see it basically down here. And there's that opening. So I mentioned the prostate gland, right? And so there are a lot of accessory glands that help with both nourishing the semen or the sperm, and to help move it through and give everything that it needs to work correctly. So let's talk about the male accessory glands, and we're going to start with the seminal gland first. So the seminal gland is on the posterior bladder surface. It contains smooth muscles that contract during ejaculation. And it's important because it produces viscous alkaline seminal fluid. Now that seminal fluid is going to contain fructose, citric acid, and coagulating enzymes. Perfect example of an enzyme that it has is vesiculase and prostaglandin. Now, when they're doing CSI television and they're trying to figure out, if there's blood or different kind of fluids on things, that sometimes they light up by UV light, and there's basically yellow pigment within it that fluoresces with UV light, and that's how they can pick up. And it comes from the seminal gland. Basically, this comprises about 70% of the volume of the semen. And if you remember, the seminal gland joins the ductus deferens or vas deferens to form the ejaculatory duct. So this is the seminal gland. The next one that we're going to talk about is the prostate gland. Now, the prostate gland will encircle the urethra that's inferior to the bladder. It's about the size of a peach pit, that inside part of a peach. It does contain smooth muscle as well that contracts during ejaculation. And this is going to secrete milky, slightly acidic acid fluid. Now, this contains citrate, enzymes, and prostate-specific antigens. A short for that is PSA. So what this does, it plays a role in sperm activation. And so this makes up about 1/3 of the semen volume. Now, there are homeostatic imbalances associated with the prostate gland. So let's talk about a few of them really quick. And some of you guys may end up suffering from this. I know I've had family members have these problems. So prostatitis is something that's actually pretty common. And this is overall an inflammatory disorder. It can be caused by bacterial infections, and so you can have acute prostatitis or chronic prostatitis with this, right? They put acute or chronic. And a lot of times what they do is they treat this antibiotics. Now, it's not just from bacterial infections. It can also be caused from injury, or if you have some kind of problem with an immune system disorder. So it's not just if you have a bacterial infection, it's just that that's one of the common parts. Now, chronic prostatitis, acute is something that's going to be limited and you can get over it pretty soon. If you kind of have this issue that remains in your life constantly, it goes into chronic prostatitis. And this is also known as pelvic pain syndrome. Now, they're both inflammatory and non-inflammatory types. So this guy has UTI-like symptoms. And you might ask yourself, what is that? And basically, if you've ever had one, you get pain in external genitalia and lower back. It can get it where like UTI symptoms, it hurts to pee. You have a hard time urinating it because it's become inflamed, so it cuts off your urethra and it doesn't go through there. And another thing that you can have is leukocytes actually in your urine. Now, the symptoms are the same as above, except there's no leukocytes or bacteria in the urine at all. So that's with chronic prostatitis. Now, there's also known as benign prostatic hyperplasia, and this may be age-related. It's basically when there's a distortion in the urethra, or this problem distorts the urethra. Sorry for my misspeaking. And so it can be treated with surgery. That's the most common type that's been used the longest, but there are newer options. For instance, they have used microwave to shrink the prostate. They've used what's known as balloon compression. And radiofrequency radiation. All of these aimed to try to allow the urethra to work, and most of them trying to shrink the prostate or make room in that area for their urethra. And the last we're going to talk about is prostate cancer. So prostate cancer is the second most common cancer or the second most common cause of cancer death in males. It's treated by surgery and radiation, castration, and drugs. They have done some clinical trials to do other types of treatment, like cryosurgery, different types of chemotherapy, ultrasound, and proton beam therapy. And only time will tell if they become really, really good and a more common type of treatment for this. And the last type of male accessory gland that we're going to talk about, the third one, is the bulbourethral gland. It's a pea-sized gland that's inferior to the prostate. You can see it right over here. And basically, it produces thick, clear mucus during sexual arousal. And that's important because it helps to lubricate the gland's penis and neutralizes the traces of acidic urine in the urethra to give the sperm a nice, safe place to go through, right? Now, what's going to go through here? Well, sperm, of course, but sperm is part of something bigger, right? Essentially known as semen. And semen is this white milky mixture of sperm and accessory gland secretions, right? And basically, whenever we have this, men basically ejaculate about 2 to 5 mils of semen. And that semen is going to contain anywhere from 20 to 150 million sperm per milliliter. So it's going to have a lot of sperm in there, right? And that semen contains what's known as fructose. And fructose is important for ATP production. It helps to protect and activate sperm, and it's going to help facilitate sperm movement. And so this stuff, basically is an alkaline fluid. Remember that you have a couple glands that are working together to make it that way. And that does twofold. It's going to basically neutralize out the urethra in males, right? And remember, we're talking about because they use both the urethra for the release of urine in combination with the release of sperm. So there's some urine that's left over there. So we want to be able to neutralize that out. But sperm is going to leave, and it's going to go into a cave unknown to any sperm before it. That's actually a lie, right? It happens over and over again. But it ends up going into a new place outside the body known as the female vagina. And the female vagina is acidic in nature. And so it's going to help to make that area more friendly to then enhance motility or movement. So all this is going to work together. Now, there's a bunch of different characteristics of semen. And we've talked about kind of all these different things that the different types of glands produce, right? And we're kind of like, well, what's the purpose of that? And so, for instance, they have prostaglandin within semen. And so this is going to do a couple things. It's going to decrease the viscosity of mucus in the cervix. And it helps to stimulate reverse peristalsis in the uterus. So it's going to allow the sperm to be able to move around better, and it's going to allow for it to be moved up through the female reproductive system. Now, it contains different hormones. For instance, one known as relaxin. And other enzymes that basically enhance sperm motility. It has ATP. That's important for energy production, right? And we talked about it comes from fructose. It has the ability to suppress the female immune system. You have to remember, when semen is going into a female's body and a reproductive system, it's considered an outsider. So the body under normal situations is going to try to protect itself and start trying to kill things. There's a really great movie called The Great Sperm Race it came out of England, and it kind of shows you what it's all about. And it uses, instead of it showing sperm through the whole thing, people represent sperm. And it kind of shows you the whole process of what a sperm has to go to actually reach the egg. And it's a feat. It's amazing. And so it's not going to get rid of the immune response the female has, but it'll help reduce it or suppress it. Now, semen also has antibiotic chemicals that help destroy some bacteria. And last but not least, it has clotting factors. Remember I said that earlier? You might have been like, this is weird. So basically the clotting factors help to coagulate the semen initially. And this helps to prevent draining out. We don't want everything just to flow out all the time, right? And then what it's going to do is it'll liquefy it by fibrinolysin, so sperm can move. So these are the 6 characteristics of semen. And so when we're talking about the male reproductive system, we have to start talking about, when we're talking about the physiology, it's going to be enacted through the sexual response. And there are 2 parts of the male sexual response. There's both an erection and ejaculation. So, erection. An erection is just an enlargement and stiffening of the penis. And basically what happens is that arterioles are normally constricted, and in sexual excitement, it's going to cause the central nervous system activation of parasympathetic nerves. And they're going to release nitric oxide. And basically, nitric oxide release causes relaxation of the local vascular. When the smooth muscle can relax, the arterioles will dilate. And basically when this happens, the corpora cavernosa expands and it retards the venous drainage. And because it retards this drainage, basically, that the blood can't leave, it's going to lead to the engorgement of erectile tissue with blood. And this is what's going to cause the penis to stiffen and enlarge. The corpus spongiosum is important because it helps to keep the urethra open so that semen can leave, right? Now, erectile dysfunction can be a problem. And basically, this happens when the parasympathetic nerve of the penis releases too little nitric oxide. And this can be because of alcohol, it can be because of drugs, hormones. There could be either blood vessel or nerve damage. And it can be a problem, and I'm going to put it up here so that you can see it on the side. It can be an incompetent venous valve that basically fails to retain blood in the penis. Now, there have been drugs like Viagra and Cialis that potentially exist to help to basically fix this problem. Now, with erection, overall, if you don't have this erectile dysfunction problem, that basically an erection is initiated by sexual stimuli, so such as someone touching or mechanical stimulation of the penis, things as much as erotic sight, sounds, and smell. And they can be induced or inhibited by emotion or higher mental activity. So basically keep that in mind. And there are basically longitudinal and circular collagen fibers that are around the penis that are preventing it from kinking or buckling of an erect penis. Now, the ejaculatory process of male response is when you have the propulsion of semen from the male duct system, and we talked about the duct system itself. And this goes from a sympathetic spinal reflex. So here, the bladder sphincter muscle constricts, preventing urine from leaving, right? Because there's never that mix. And basically, the duct and accessory glands are going to contract and empty their contacts into the urethra. And you have the bulbosponginosus muscle that undergoes a rapid series of contractions that's going to cause the expulsion of the semen, right? So this is a muscle there that actually allows for it to leave. And it's closely to 11 miles per hour, right? And so the ejaculatory event itself is called climax or orgasm. And I have already talked about this, so I'm going to move past that. So let's go on really quick. We talked about inside the semen, there's sperm. But how do we actually produce the sperm? How do they get made? The process is called spermatogenesis. And spermatogenesis is the actual formation of the sperm, And you can think genesis as if you think of beginning, and you see sperm in that word. Now, they're also called spermazoas, so don't get confused on that. And this production actually happens in the seminiferous tubules, and it takes between 64 and 72 days. Now, sperm are interesting because they only incorporate one set or half the set of chromosomes that everybody has. So most body cells have 46 total chromosomes. There's 2 sets of 23 pairs of chromosomes, right? Gametes only have 1 pair. So this up here is what's known as a diploid cell. And basically these gametes or sex cells are known as haploid. And you can tell the difference. A haploid cell is 1N and a diploid cell is 2N. Okay? And so this whole process of spermatogenesis, that's specifically for the production of sperm, uses a process known as meiosis. And I like to think of meiosis as making little me's. And so basically what they do is they take these diploid cells, regular body cells, and they cut them in half, right? So you go from 2N to 1N. And this is going to help induce genetic diversity, as all daughter cells, or those are your offspring cells, are genetically different from the original cell. So let's talk about meiosis, and this is going to be a video that helps to introduce it.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
So that kind of gave you an overview on what we're going to talk about. So mitosis we hit on in A&P 1 towards the beginning of the semester. And mitosis is a way to basically allow cells to reproduce when we're growing or to help replace when they become old or damaged. And so basically what happens is you have a mother cell or the initial cell, and basically what happens is they duplicate the number of chromosomes and they divide once. And basically from there, you get the exact same cell. That should make sense whenever you're trying to replace skin cells or some other type of cell like that. Now, how that differs from meiosis is that you still have that duplication process at the beginning. That happens during the S phase of interphase. And then you actually have the process of meiosis happening, where essentially you are going to separate all the homologous chromosomes are going to separate, right? Here, you can see that you have the homologous chromosomes together, and basically they separate into sister chromatids. They move apart. Where here, the very first division is just the homologous chromosomes. So when we come to, and that's what's known as mitosis 1, or meiosis. I'm sorry about that. In the second part, you're going to get the division of the sister chromatids. And you can see from here that each one is by themselves. And this is what essentially is going to pick up the gametes. And at this point, we have the haploids, or N. So this is going to have 2 divisions, where this one has 1 division, right? This is what's known as meiosis 2. So this is the main difference between the mitosis and meiosis, is that you start with a 2N cell. In mitosis, you end with a 2N. In meiosis, you end with a single cell. And you have 2 divisions in meiosis. We also have something known as crossing over that happens in meiosis. That doesn't happen in mitosis because the homologous chromosomes, the set that looks like your mom and the set that looks like your dad that look similar, they never come together in mitosis. So let me really quick show you and do a comparison between meiosis and mitosis. Since you should already know mitosis, if you don't, then you should go back and review it really quick, because the stages or what happens a lot have similarities between that and mitosis and meiosis.
Prior to meiosis or mitosis, DNA replication occurs. In both meiosis and mitosis, the nuclear membrane breaks down as the DNA organizes into chromosomes. In meiosis, chromosome pairs come together, or synapse, and crossing over occurs, resulting in mixing of the genetic information between the chromosome pairs. The paired chromosomes then align along the central plate of the cell and subsequently separate, one traveling to each end of the cell. In meiosis, a second division sequence occurs, resulting in 4 cells with half the number of chromosomes. Mitosis involves a single division sequence, resulting in 2 cells with no net change in the number of chromosomes.
So hopefully that helped you to kind of understand the difference between the two. So let's actually talk now what it is. So meiosis 1, this is going to be the very first separation. So you're going to have within meiosis 1, and you always have to put the 1 behind it. That's telling everybody that you're in the first division. The first part of meiosis is actually prophase 1. Now there is a stage right before that's called interphase. Interphase is a preparatory stage. It's not part of meiosis. And basically it has 3 different stages. It has gap 1, the S phase, and gap 2. And basically the G phase, what's happening there, both of them, is that you're having growth of the cell. This is where the cell becomes double the size. You're getting more organelles. All of those activities are happening in G1 and G2. During synthesis, this is where DNA replication happens. And we talked about DNA replication last semester. I'm not getting really big into this because this was already covered in part 1 of this course. And again, if you don't remember this, then you need to go back and search through that material and just get brushed up on this. And so once we get done with that interphase, we go into meiosis itself and prophase 1. Now, prophase 1 here, just like in mitosis, all those same things are going to happen. You're going to get the nuclear envelope is going to start disintegrating. You're going to have the spindle or the centrioles go to opposite sides of the pole. You're going to have the chromosomes start to condense. So all of those things that you remember from mitosis is going to happen in for prophase are going to happen in prophase here as well. The difference here, a big difference, is that you get the homologous chromosomes that are going to be pairing. So the chromosomes that you have from your mom that have the same, like chromosome 1, is going to pair with chromosome basically 1 that your dad gave you. And because they're so close to each other, they're attached, they're going to have what's called crossing over. And this is where basically genetic recombination is important. And so here you can see it. Here are the homologous chromosomes. They look the same. The red is from mom, blue is from dad. And you can see on each one of those you have sister chromatids coming together. So they're going to form what's called a synaptic complex here, right, on the ends. And this is basically where the synapse begins. These are what are known as triads or bivalents, right? Triads because there's 4, bivalent because there's 2 of these coming together. Now at this point crossing over can occur, and you can see that's when you take a bit of genetic material from mom and a bit from dad and you flip them together, and that's what's known as a chiasma. You can see this here. And so here's a picture of what a chromosome looks like, and you can remember the kinetochores here. That's the area that basically is going to be on the outside of the centrosome in the middle. That's going to connect to the microtubules when they move those around. Some of this I'm assuming you know because it's been taught in part 1. And so from here, you can see a chiasmata here and here. And so because this can happen, you can see the arrangements of these sister chromatids here can be quite different between offspring. And this is the reason why not every single offspring looks exactly the same. Because if you look, this is the original that you have for your dad. This over here is the original from your mom, right? If you look here and here, these are completely new, right? You have all the dads with a section of mom on there. So imagine if you get the arrangement of different possibilities you can have with crossing over, and then when you actually go to line these up in the middle, that the way they line up here is independent. Each chromosome pair that comes to the middle is independent of the other. It's called independent assortment. So they can line up all differently in the middle. So what kind of chromatids that you have in each gamete can be drastically different. So this is what meiosis looks like. So here's interphase 1. Oops. And you can see nothing big here, but prophase 1, this is mid, and late. You can see that the nuclear envelope is gone, and you can see these homologous chromosomes are pairing up together, right? You're going to have the centrioles going to opposite sides. You can see them here, right? And you're going to start getting the development of the spindle fibers happening. That's what attaches to the chromosomes itself. And so from here, metaphase, anaphase 1, the exact same thing happens as in mitosis. And so let's look at here, metaphase 1. You can see all this homologous chromosomes are meeting in the middle, down the midplate. This is a part where the chromosomes are actually the most condensed as well. Here, the homologous chromosomes are separating. So you get one set here and one set here, right? So they're pulling apart. You can think M, middle, A from away. And basically, telophase is the opposite of prophase. So except for the, the crossing over and all that, All the other things that are happening in prophase, basically the opposite is happening here. So the nuclear envelope's coming back. The chromosomes are uncondensing. You have the centrioles are now together. The spindle fibers are gone. So everything that's happening at the beginning of prophase or through prophase, the opposite is happening in telophase. And if you notice now we have 2 cells. So number 1, for the most part, if you see 1 cell there, you can think you're in meiosis 1, all the way till you get to the very end of telophase. Now within telophase, you have what's known as cytokinesis, and this is the separation of the cytoplasm. Meiosis 2, this is when you're taking the sister chromatids that are together and separating them out, right? Now each form of chromatid is now going to be their own chromosome. So if you see, all the same things happen in prophase as before, and you can see that they're starting to condense. And this is prophase 2, and of course you can see 2 cells. They're meeting in the middle, and again, you see 2 cells. There's just a separation in the middle of the 2, but they kind of go together. Anaphase 1, M for middle, A for away. Here you're having the sister chromatids separate into opposite sides. So before you had 2 chromosomes together, and one would go this way and one would go this way. Now these guys are separating, and they form chromosomes on the opposite poles. And then at the end of this, for telophase 2, you're going to see, oops, I'm sorry guys, 4 cells here, right? Of course, cytokinesis is happening here. And these, you're going to go from 2N to now they are haploid 1N cells. So that was a video that kind of showed you the whole process all the way through for both. Now, when we're going through the process of meiosis, there is this idea that's called independent assortment. And what it means is you have each set of homologous chromosomes line up in the middle, right? And the orientation that they line up is independent of the other. So you just see how this one over here has both green on one side and blue on the other? Well, this guy easily could be the opposite. And if that happens, you can see how the combination of daughter cells changes. And then imagine this 23 times. So it does allow for this genetic diversity in addition to the crossing over. And so basically this is just the whole set going all the way through, and I'm not going to spend much time because I've shown a couple videos now and I've talked, but you can look in your book and get a really good idea of the separation and how meiosis and 1 and 2 differ from each other. Now when we're talking about spermatogenesis itself, this is a process that happens in the seminiferous tubules. And so this is where the spermatogenic cells are going to give rise to sperm, and it takes about 64 to 72 days. And there are these basically overall steps to spermatogenesis. The first one is mitosis of spermatogonia. These are the stem cells, and these are going to be what forms 2 spermatocytes. At this point, the spermatocytes will undergo meiosis, and they will form secondary spermatocytes, which then will turn into spermatids. And the spermogenesis is the process of spermatids, which are not active sperm, becoming the spermatids coming sperm, right? Don't get the spermogenesis confused with spermatogenesis, which is the entire process of it. So let's look at a picture and talk about how it works. So let's look over here. And basically this testes, particularly what's happening in the seminiferous tubules. So you'll notice here, right? That is what's called the basal lamina. And attached to that are these things called spermatogonia. These spermatogonia are the stem cells. And basically right here is where mitosis happens. So you can see these line up with each other. So right now, this guy right here is this guy right here. So they're going to undergo mitosis. And they are going to divide basically into daughter cell A or daughter cell B. Now, this whole process, or spermatogenesis, essentially begins at puberty. It doesn't happen early on in life. And basically, this starts with the spermatogonia, right? Again, these are stem cells. And so what happens is when you become daughter cell A here, right, you remain onto the basal lamina, and you are going to end up being a germline. This is how they keep constantly being able to have those cells, because the cells will get old. And so they divide. One stays at home by that basal lamina to continue the process. The other is going to go down, and this is the B cell line, right? And this is going to move towards the lumen, and this is what's going to develop into what we know as the primary spermatocyte, right? So at this point, and you can see here, this is A, this is B, and this is the primary spermatocyte, right? If you notice right here, you'll see there's a tight junction during that process. Do you see that guy right there? We're going to talk a little bit later about these tight junctions, but just to let you know, they form blood testis barriers. As I was telling you earlier, sperm's not formed until puberty, right? So it's actually absent during the immune system development. So this guy right here is going to prevent sperm antigens from escaping into the blood and causing activation of the immune system. So if sperm aren't there when the immune system's developing, the body won't be able to recognize it as a self cell. So it would essentially be attacked. So to avoid it from being attacked, there are these tight junction forms there to basically prevent it from escaping into the blood. Now, so that's mitosis, right? As you can see, we originally had the 2N cells here, right? And we're still at 2N. And at this point, once we get the primary spermatocyte, that guy is going to undergo meiosis. So here we get the primary spermatocyte, which is 2N, and he's going to undergo meiosis 1. And that is going to basically form 2 secondary spermatocytes. So you see here, meiosis 1, so you're going from N, 2N to N, and these are the secondary. And during that time, right, you're going to also, right after that, you're going to get your second division, right? So this guy is going to divide here and here. So each of these secondary spermatocytes are rapidly undergoing meiosis 2 to become 2 spermatids. And you can see here, these are early spermatids, right? And mitosis 2, or meiosis, I'm sorry. So this is where we get spermatocytes changing to spermatids, and this is meiosis. So this top part, mitosis, the second part here is meiosis, and the last part we're going to look at is the spermiogenesis, which is taking these spermatids and turning them into sperm. And so basically what's going to happen here, they all have the correct number, N, or the haploid number, that they need for fertilization. These guys, though, aren't able to move. So they're unable to swim, but the pressure of the testicular fluid pushes the immobile sperm into the epidemias, where they can gain motility and fertilization power, right? But during this spermiogenesis, basically it's a streamlined process where the spermatids are going to elongate. You see how they go from being just circles and then they become elongated. They're going to basically lose cytoplasm. They're going to form tails. And they're going to go into these 3 different regions, right? So this is the last part, and you can see it over here as well. So this is this process of spermatogenesis. Now let's look really quick at spermiogenesis so that we can just take a quicker look at it. This takes approximately 24 days to happen. And as you can see, this is the earliest spermatid, right? And essentially, you can see over here, the centrioles disappear, and you can start seeing the microtubules helping to form the flagellum. And basically, these mitochondria will line up towards that middle part. It'll form the chromosome here, and that is one part. This is part of the head region, right? And this is what's going to have the genetic material. That's going to basically have the nucleus. And this chromosome here, it basically has the hydrolytic enzymes. Those hydrolytic enzymes, if you remember, they help the sperm to penetrate the egg. Excess cytoplasm is leaving there, and you can see it's starting to form in that middle area, the midpiece. This is the metabolic region. So that's going to produce all the ATP. As you can see, the mitochondria are lined up there. And the last part is this tail region. And that has a flagellum, and so it's for locomotion or movement. So this is the process of making functional motile sperm. Now there are supportive cells there, and they're called Sertoli cells, right? And they have several different functions. So they extend through the walls of the tubules, and basically they surround developing cells. They provide nutrients, and they help signal to dividing cells. Essentially, the cells that won't make sperm. They help to move cells along to the lumen. They secrete testicular fluid into the lumen that help with sperm transport. They help to phagocyze faulty germ cells, and all the excess cytoplasm. Don't need stuff, so they get rid of it. And they produce chemical mediators. These guys are going to regulate spermatogenesis. And again, they contain tight junctions that help divide tubules into two compartments. These tight junctions, again, prevent sperm antigens from escaping into the blood and causing activation of the immune system. And that's because sperm is not formed until puberty. So it's absent during the development of the immune system. And so the body can't recognize sperm cells as being part of its own body cells, so it will attack it. So it's a way to separate it so that doesn't happen. You have the basal compartment. This is an area where the spermatogonia and the primary spermocytes are located. And the abluminal compartment is the area where metabolically active cells and tubulins are located. And so there's a way that we regulate the reproductive system, and that's through the hypothalamic pituitary gonadal, or HPG axis. And this is for the production of gametes and sex hormones, how they're regulated by the sequence of hormonal events involving the hypothalamus, the anterior pituitary, and the testes, right? And it involves these set of different hormones. You have the gonadotropin releasing hormone, you have the follicle stimulating hormone, you have the luteinizing hormone, you have testosterone, inhibin. So all of these work together. Now I kept these here so that you can read through them. But I'm going through and we're just going to actually look at the picture as we go through here. So the very first thing that happens is that the hypothalamus is going to release GnRH, which is the gonadotropin releasing hormone. And so this is going to go from the hypothalamus to the anterior pituitary gland, which essentially then going to release follicle stimulating hormone and luteinizing hormone. The follicle stimulating hormone is going to go down and it's going to stimulate spermatogenic cells to start producing sperm. And it does this indirectly by stimulating those cystinocytes to release antigen binding protein, otherwise known as ABP. And so ABP keeps concentrations of testosterone high near spermatogenic cells, promoting spermatogenesis, and that's how it works. Now, the luteinizing hormone here is going to bind to interstitial endocrine cells. And that's going to prod them to secrete testosterone. Rising testosterone levels, right, are basically going to stimulate sex organ maturation, develop and maintain of secondary sexual characteristics and libido. All these things we're going to talk about here in a second. Now, testosterone or rising levels of testosterone is going to lead to feedback, though, of the hypothalamus. And once those are high enough, right, so you're going to see that it increases spermatogenesis. It helps with primary and secondary sex characteristics, primary being the actual sperm, secondary being all the things that you see on the outside. But then high testosterone levels will lead to negative feedback. So testosterone will come back to the hypothalamus and inhibit GnRH production. And inhibit basically the pituitary system from releasing basically GnRH, and that will stop the FSH and LH, or follicle stimulating hormone and luteinizing hormone. Now, there's also something known as inhibin, which basically is released by cystinocytes when sperm count is high. And basically, this inhibits this and this. So this is how basically sperm or spermatogenesis is regulated. The amount of testosterone in sperm produced by testes basically reflects the balance among the three interacting sets of hormones, specifically the gonadotropin releasing hormone, follicle stimulating hormone, and luteinizing hormone. The balance takes about 3 years to achieve, after which testosterone and sperm production are fairly stable throughout life. Without the gonadotropin releasing hormone and gonadotropins, testes will atrophy and sperm and testosterone production ceases. Before birth, male infants have testosterone levels about 2/3 of adult, but soon after birth, the levels will recede and remain low throughout childhood until puberty. And so this is basically showing the basically levels of testosterone. You can see at fertilization they're high, but during those basically from about 3 to about 5 months that it peaks, and then it slowly comes down till birth. It peaks really quick at the beginning, and basically, and then once you hit puberty until adulthood, it gets really, really high until about the age of 60, where it starts falling again. Basically, testosterone comes from cholesterol. It looks very, very similar to it. So basically, cholesterol, when you look at it, it ends up helping to form both estrogen, or cholesterol is used to form both testosterone and estrogen, obviously. the two main sex hormones that are in male and female. It can be converted to what's called DHT or dihydrotestosterone in prostate and estradiol in some brain neurons. This helps to promote spermatogenesis and targets all accessory organs. It can have multiple anabolic effects throughout the body. Deficits can lead to atrophy of accessory organs, semen volume declines, erection and ejaculatory arc can be impaired, and basically treatment is used for testosterone replacement. Okay, so when we're talking about testosterone and its effects, the primary sexual organ that we're looking at is essentially the gametes. But testosterone can also affect what's known as male secondary sexual characteristics. And these features induced in non-productive organs by male sex hormones, mainly testosterone. And basically, think about it. It's an easy tell-all way for the body to show an outward way of what type of gametes you have. Imagine if everybody looked like a male or a female, how hard it would be to determine which person you would need to pair up to reproduce. You would be going to basically all individual people and going, hey, do you have this? I have this. And they'll be like, well, I can't reproduce you. Do you have this? I have this. Oh, no. So these are ways for which we can basically physically see if someone has the male primary sexual characteristics. And I'm going to have to, here, thank goodness. So let's see what those are really quick. So these basically features induced in non-reproductive organs by male sex, so mainly testosterone. So we have the appearance of basically pubic, armpit, and facial hair. We have an enhanced growth of chest hair. Deepening of the voice. Thickening of skin, and it becomes oily. Bones grow, and they become more dense. Skeletal muscles increase in size and mass. You have a boost of basal metabolic rate. And this is basically, I'm going to add a little 8 in here. It's the basis of sex drive or libido in males. And it is more complicated, I hate to say. And it's more complicated because of pregnancy. And so we've already talked a little bit about this, so we're just going to review some of this. So ovaries are basically the female gonads, and they produce the female gametes. Let me change colors here. And do you remember what gametes are? They're the sex cells, right? And so the female gamete is an ova. It helps in secreting female sex hormones. So can you name what the two sex hormones are? Hopefully you put estrogen, which other forms is estradiol, estron, or estrol, and progesterone. And they also have a duct system as well. You have the uterine, known as the fallopian tubes, the uterus and vagina. And the last is the external genitalia. And just to let you know, just like in the men's, we're going to start with the external genitalia first. And so the female external genitalia, also called the vulva or pudendum, includes the following. You have the mons pubis, which is that fatty area overlining the pubic symphysis, and you have the labia majora, which is that hair-covered fatty skin fold. Now, you do have a counterpart to the scrotum. And basically that's incorporated into the labia minora, which is the skin fold that's lying within the labia majora, and this vestibule area here, which is essentially the recess within the labia minora. Now, if you look, other parts on here include the fourchette. And this fourchette is a ridge that's formed, and basically it's formed by joining of the posterior vestibule and the labia minora. Now, if you look over to the right or left of that, you'll see what's known as the greater vestibular gland. And this is homologous to the bulbourethral gland. And basically, this guy helps to release mucus into the vestibule for lubrication. Now, if you look over here, we do have the clitoris, and that's just anterior to the vestibule area there. And what you have is on here, it shows you the glands of the clitoris. So this is the part that's exposed. And the pubis of the clitoria is what hoods the glands. Now these are the counterpart basically of the penis. And then if you look at this diamond shape here, if you remember in the men's, it's the same name, perineum. And basically it's this diamond shape, if you remember, between back here is the coccyx, right? You have the pubic arch over here, and then you have the two ischial tuberosities. And it makes that nice kind of diamond shaped area. So that kind of gives you an idea of the external genitalia. Now, for the ovaries. Within the ovaries, you have follicles, and the ovaries house these mini ovarian follicles, which are sac-like structures. Basically, each follicle consists of an oocyte, and so this is an immature egg. And it's basically surrounded by follicle cells or granulosa cells. So it's follicle cells if only one layer is present. And basically, if there's more than one layer present, then it's granulosa cells. And basically, these follicles go through several stages of development. They start with what's known as primordial follicles. And so this is basically a single layer of follicle cells, and I'm just going to put FC here, plus oocytes. Now, as this becomes more mature follicles, what do you think will happen? Well, if you get more mature, basically then you're going to have several layers of what? Granulosa cells plus an oocyte. So it's going to transition. From here, you're going to have what's known as a vesicular, or it's known as an antral, or actually tertiary, I'm just going to put a 3 there, follicle. And this is basically a fully mature follicle. And basically there's a fourth here. And this, you end up having a fluid-filled antrium forms. And basically what this is that this is the point where the follicle bulges from the ovary surface. And you can kind of go up here and look at this top picture where you can just see it happening. So here you have that primordial follicle, you have that oocyte, and you just have that single layer that's going around it, right? And then basically after that, you can see that there's a growing follicle here. So this is going to be a more mature follicle that starts having all these granulosa cells forming around it and the oocyte. Now, if you get over here, you're having that third, the vesicular follicle. This is fully mature follicle, and it's almost ready to burst out. And this is basically, you can see it's starting to bulge here too. That's when you're getting into that fluid-filled antrum forms, where it starts to basically bulge. So this is the antrum, if you see right there, right? And so that kind of gives you an idea of what's happening and what it looks like. And then it finally releases here during ovulation. And so as we talk about the ovaries, they're actually put into place by several different ligaments. And so here we have, oops, I'm sorry guys, the suspensory ligaments. And these secure the ovaries to the lateral wall of the pelvis. You have the ovarian ligaments. These anchor the ovaries to the uterus, and then you have the broad ligaments, right? And these are folded the peritoneum that's enclosed and holds the ovary in place. And realize the broad ligaments, that's a broad term because there's several different ones, but we're not going to get into each individual, but I will show you on a picture what they look like later. Now, the blood supply for the arteries are from both the ovarian artery and the ovarian branch of the uterine artery. So it gets a blood supply from two places. And basically each ovary is surrounded by fibrous tunica albuginea, which is then covered by a germinal kind of cuboidal epithelial layer. And basically this germinal layer is continuous of the peritoneum. So that kind of tells you a little bit about the composition of the ovaries and how they're held into place. Now, when we're looking at the ovaries itself, it's made up of a couple of different regions. You both have, if you see here, the cortex or this outer cortex, which this is what houses the forming gametes. And then if you look here towards the middle, just think this is kind of how everything we've looked at. If you've looked at the lymph nodes or if you looked at the kidneys, they always have the outer cortex and the inner medulla, right? And so this inner medulla is the part that contains blood vessels and nerves. And you can see that in there already. Now, what's going to happen in here is this oocyte is going to become a mature egg and then be released. And the ejection of the oocyte from this ripened follicle is what's known as ovulation. And basically, once the oocyte is ejected and becomes the ova, what's left of that follicle is what's going to end up turning into what's known as the corpus luteum. And this process, on average, occurs about every 28 days. Now, the female duct system, let's get into this. The uterine tube system does not have direct contact with the ovaries. The ovulated oocyte is cast into what's known as the peritoneal cavity, where some oocytes never make it to the tube system at all. And then if they do, the tube system will include the uterine tube, the uterus, and the vagina. And so let's actually talk about the different parts of the tube system. And we're going to start with first the uterine tube, also known as the fallopian tube or the oviduct. Now, this receives ovulated oocyte and is usually the site of fertilization. This is not the site that the cell or the potential zygote and offspring become attached to, right? This is just where fertilization happens. Now, there are different regions within the uterine tube. The isthmus, the ampulla, and infundibulum. which are the three parts I'm going to show you here in a second when we look at the actual uterine tubes. Right? And so when we're talking about what they're important for, the isthmus is basically this constricted area where the tube joins the uterus. The ampulla is on the distal end of the tube that curves around the actual ovaries. And the infundibulum is the distal expansion that's near the ovaries themselves. Now, within this region, they basically have these ciliated fimbrias. And these are going to help create current to move the oocyte into the uterine tube. So basically, these guys receive the oocyte from the ovary. The cilia are basically located inside the uterine tube. And basically, they're going to help to slowly move the oocyte towards the uterus. And this normally takes anywhere from 3 to 4 days. So the first one basically takes the oocyte from the ovary and gets it into the uterine tube. The second one, basically once it's in the uterine tube, it's going to move the oocyte to the uterus. Now the uterine or fallopian tube has a few functions. So one, it basically receives the ovulated oocyte, right? And it receives it from the ovaries. It provides a site of fertilization. It empties into the uterus. It basically has little or no contact between the ovaries and the uterine tube. And it's basically supported and enclosed by broad ligaments. So I can almost say these are characteristics more so than function. Let's see. I think is a better word. And so, that being said, let's look at a picture a little bit. And so let's go and talk about the different parts first of the uterine tube. And so you can see it over here. So here is our ovaries. And the fimbria are these little things here. Remember the finger-like structures that help to get it into this area. The infundibulum is that first area that's here that incorporates these guys to help get it in. As you can see from there, it goes to ampulla that goes through here. And then this area right close to the opening or the lumen of the uterus is the isthmus. Right? And so that kind of gives you all the different areas here of the uterine tube. Now you can see basically that it's held in place by 3 main ligaments. You can see the suspensory ligament here. You can see the broad ligament here. And if you look, there's several different ones, but we're not really looking at those in particular when we go through here. The only one that I will point out is the mesosalpinx. And basically this, the reason why I put is a little bit different because it is short mesentery that's there versus a typical ligament. And then you can see the ovarian ligament down here. And so these external uterine tube are covered by the peritoneum and again supported by that mesosalpinx. And so that kind of shows you the uterine tube. Now, there are some homeostatic imbalances associated with the uterine tube. First of all, there's an ectopic pregnancy. And so this is when the oocyte is fertilized in the peritoneal cavity or that distal uterine tube and begins developing there. Normally aborts naturally with substantial bleeding, and it's a lot of pain. And if it doesn't terminate naturally, there is a surgery that has to happen. This can kill you, just to let you know. So it's not something that you want to happen. There's also pelvic inflammatory disease that's PID, that's a result of this, and it's a spread of infection from the reproductive tract to the peritoneal cavity, and may cause scar tissue and lead to infertility. Now, you see on like packets of Tampax that this is always a warning. And it's kind of interesting because it still happens today, but it's not as common as it was, say, in the early 80s. In the early 80s, when they made tampons, they actually made this absorbent material. They no longer use it anymore, but they made this absorbent material that was so good at its job that women were using it all day long. Like, they weren't changing routinely. But the problem is that it would absorb water and solution so well that it would dry out the layer inside of your uterus and vagina. And so what would happen is they would pull it out and they take the top layer of that tissue with them. And then they put a new one in there. And then that tissue would get infected. And then that would lead to a spread into the uterine tubes and PID or PID. So they started warning people of it. They stopped using that kind of absorbent. And that's kind of where all those warnings came from, because women did die from that. And so just to give you kind of a little history on both those homeostatic imbalances. The next part of the duct system is the uterus. And so this is a hollow, thick wall, basically muscular organ. And its function is basically to receive, retain, and nourish a fertilized ovum. Right? And so there's a position that the uterus can have. It's either anteverted, which is inclined forward, and that's the normal position, or retroverted, which is inclined backwards, and that can basically make having a baby extremely difficult. Now, we're just going to talk about this, but I'm going to show it on a slide here, a couple slides forward from here. But they do have the region of the uterus. There are different parts. There's the body, which is the main part. There's the fundus, which is the superior rounded region that's above where the uterine tube enters. And think rounded superior region, just like in the stomach, right? And then the cervix, which is the narrow outlet that protrudes into the actual vagina. And the last part is the small part that's called the cervical canal, that's going to essentially communicate with the vagina via external OS and then the uterine body. And so that's the different parts. Now, it does have a cervical gland, and this gland helps to secrete mucus that will actually block sperm entry during any time except during the mid-cycle. And we're going to talk more about that. Now, there are different walls of the uterus. You have the endometrium, which is basically the mucosal area. And so this is the inner lining, right? And this is what allows for implantation of the fertilized egg. This is what's going to slough off if no pregnancy happens, right? Just going to put period here. And that's what's called menses. Myometrium is the middle layer. And this is basically smooth muscle. And this is what contracts during labor. Perimetrium is the visceral peritoneum. It's the outermost serous layer. So that's basically talking a little bit about the uterus. So let's look at the uterine wall now, and specifically look at the endometrium, because this is the part where that's going to support life, and this is the part that we lose during menstruation. And so the endometrium actually has two chief layers. These layers are otherwise known as strata. And so they're the stratum functionalis, which is a functional layer, and the stratum basalis, which is the basal layer. So the functional layer actually changes, in response to ovarian hormone cycles. So this is what gets shed during menstruation. Now this basal layer here is what basically forms a new stratum functionalis after menstruation. So it's unaffected by the hormones. And so you can see both of them here, right? Here's the functionalis, and here's the basalis here, right? And it makes sense. This part is the part that stays behind. This part up here leaves. So how would we remake this? Well, he could, since he's there, can help remake it. Now, if you see underneath all there is that myometrium, and remember that's that smooth muscle layer that helps to contract during pregnancy. So the uterine wall has vascular supply. The way that the vascular or the veins are put together help to supply or plays a key role in the cyclic changes. So just to let you know, the uterine arteries that help supply the blood and the nutrients for the uterine wall arise from the internal iliac. Right? And basically what that's going to do is it branches into a few types of arteries. You can see here, this is the uterine artery that comes from the internal iliac. And then you can see that this branches here into the arcuate artery. And basically, then that guy is going to branch into, if you look here, the radial artery. Now, the radial artery will branch further. You'll see it goes and branches into the straight and the spiral arteries. Now, the spiral arteries are important because basically they're located now, if you look, we are in the functionalis here, right? Where the straight arteries are in the basilis. So these guys, right, are going to help with providing nutrients for those cells that will later make the stratum functionalis. But these spiral arteries are important because they degenerate and regenerate. And their spasms are what cause shedding of the functionalis layer during menstruation. Kind of cool, isn't it? So these guys, extremely important. Extremely important. So now let's just look at some of those things that we've talked about in the uterus here. And so let's look at these different regions again and just reiterate their importance and what they are. So basically the first thing we're going to do is look at the body. And you can see it's that big, large layer, and this is the main portion, right? And then if you look at that top layer, that's that superior rounded region. And if you look, this is where the uterine tube, the fallopian tubes, actually enter. At this point, you can look down here. This is the area that you have the cervix, and can you see how it's starting to narrow down and protrude into the actual vagina that's here. And then if you look at this last part right here, you'll look at the cervical canal. And that cervical canal is basically going to help in communicating with the vagina and the uterine body together. And you can see this isthmus that is there and located as well. And if we're looking at the different types of ligaments that are here that help to support the uterus, you can see those here, right? You have the mesometrium, the uterosacral ligament, the cardinal or lateral cervical ligament, and you have the rounded ligament of the uterus. So all of those four guys play a role in helping to keep this guy into place. I want to call him a guy, that's kind of funny. And then if you look here, they show you the different walls, right? And so you can see on the inside is the endometrium, then that smooth muscle right there that helps in labor contractions, and the peritoneum on the outside. Now, there are homeostatic imbalances for the uterus. And a big one is cervical cancer. It affects about 450,000 women worldwide each year, killing half. It's most common between the age of 30 and 50. And basically the risks are frequent cervical inflammation, STIs, which you might think of as STDs, and one of the most is HPV. Even multiple pregnancies can give you a higher risk. Now, there are vaccines provided. 3-dose vaccines, they can help protect against HPV, and they're recommended for about 11 to 12-year-old girls. Now, how you can detect this, and this is the reason why it's so important to go and get them, are pap smears. And basically, they're recommended every 3 years for ages 21 to 30, and every 5 years for ages 30 to 35. And basically included within our HPV test. You can discontinue a lot of time at 65 or after hysterectomy or with sexual inactivity. I would even say these are the recommended and what they state in the book, but I suggest every year, especially if you're sexually active with more than one partner, because HPV is so prevalent. Some of the stats say up to 92 to 94% of the population has contracted HPV at some point in their life. And the problem with it is that it doesn't necessarily have any physical indication that you have it. There's not even a good test. There's not a test for men to show that they have it. And a lot of times men will have no signs at all. And the sign that most of the time women have is when they go and get their pap and they get tested for STDs and they put in for HPV, or they do the pap smear and the pap smear will come back abnormal. They'll do an ELISA test to then see if you have the particular virus associated with it. And they'll do additional kind of microscope assays to look to see if you have it. So, I mean, it's a big deal, right? Now, this is not the only homeostatic imbalance. Basically, an unsupported uterus may sink inferiorly until the tip of the cervix protrudes through the external vaginal opening. And what this is known as a prolapse of the uterus. It can be caused It can be caused by overstretching and sometimes tearing of the muscle during childbirth. Even though the uterus has many agrene ligaments, it is principally supported by muscles of the pelvic floor, which are prone to damage, chiefly the urogenital and pelvic diaphragms that help hold them up. And so basically this is the side view that we have looked at, and you can see that you have the different areas. So we started with talking about the ovaries that you can see here, right? And then the little finger-like protrusions that help grab the ovum and take them into the uterine tube, right? And basically you have that first part, the infundibulum, right? And then it goes through till you get to the, basically, do they have all the parts on here? It's hard to see. They don't have them all on there until you get into that first part, right? Which is called the ampulla. and then it goes farther down, right? And then that third part right there is the isthmus, right as it enters. And so then you can see down here the uterus, and you can see it being basically supported here. You can see the round ligament. Over here, you can see the uterosacral ligament. You can see up here a suspensory ligament of the ovum. Trying to see if you can look over here, the cervix here towards the end of that, the covering, the peritoneum that's up here. We've talked about the outside with the clitoris. The uteral orifice is right here. And so you can see there's a difference between where we urinate, or females urinate, and where a child forms, or, you know, where intercourse happens, where men it's the same. Now, if you look here, there's that small thing there called basically a hymen. And this trying to see is just kind of this mucosa that is near the vaginal orifice or opening, and it's this incomplete partition, and then this ends up getting broken during intercourse. Of course, the small and the major, or minor, minora and the majora labia that we talked about earlier. Here's the vestibular gland, that's like the vulva urethral gland, right? And the next thing that we're actually going to talk about, just kind of hit some of these, is the vagina. So let's talk about that really quick. So this guy extends between the bladder and the rectum from the cervix to the exterior, and it serves as the birth canal. In addition to that, it allows for passage for menstrual flow, and it's the organ of copulation. Now, it does have several different layers. It has an eventita. It has a muscularis that's incorporated of smooth muscle. And then it has a mucosa, and it has these roots, remember, that help to basically these folds. Vaginal secretion, pH changes throughout your life. It's acidic in adulthood, and it's alkaline in adolescence. Now, like I said, there is what's known as a hymen. And basically, the hymen is an incomplete partition. And this guy gets broken during intercourse. Now, it's not just during intercourse. If you end up using tampons or anything like that, that can break it as well. Now, within the mucosa, there are dendritic cells. And it's thought that they might help to provide a route for HIV transmission. Now, because the uterus tilts away from the vagina, attempts by untrained person to induce an abortion by entering the uterus with a surgical instrument can actually puncture through the posterior wall of the vagina. So you want to make sure to have someone who is trained doing those kind of operations, because it can end up killing somebody. It can cause hemorrhaging, and if the instrument is on sterile, it can cause what's known as peritonitis. Now, there is an area called the vaginal fornix, and this is the upper end of the vagina, and this is surrounding the actual cervix. And so just to kind of show you where those are locations, so you can see there the vagina is that major opening. Towards the top where the cervix is that fornix, and then towards the bottom area, kind of protecting it, is that hymen. Now, another part that we didn't really have to discuss for men are the mammary glands. And these mammary glands are basically modified sweat glands, and they consist of about 15 to 25 lobes. And the lobules within these lobes contain granular alveoli, and these guys produce milk. And basically the milk is passed into the lactiferous ducts and then into the sinuses that open to the outside at the nipple. These are both present in men and women, but they're normally just functional in females. Now, in the female, if they're non-nursing, these glandular structures are undeveloped. Now, around the nipple is the areola, and this is the pigmented skin that surrounds the nipple. And basically, you have suspensory ligaments that attach to the breast to the underlying muscle to keep the mammary glands in place. So we can look at this really quick, right? And so you have these big lobes here, right? And within them, do you see all those circles? Those are the lobules that contain the alveoli. Now, when it goes to actually make milk from there, they get made in there and they pass into the duct, right? Into this area, the sinus. Now you can see that you have the pectoralis muscle here, and that basically these lobules and all this are attached to that muscle here through the suspensory ligaments. So that's what the lactating mammary gland structure looks like. Now there is a homeostatic imbalance for breasts as well, and that's breast cancer. Now, sorry, I'm looking, I'm missing a section. Okay. This is an invasive breast cancer. That's the most common malignancy. It usually arises from the epithelial cells of the smallest ducts that eventually metastasize, which moves to the rest of the body. It is the second most common cause of cancer death in U.S. women. So the risk factors are early onset of menstruation and late menopause. No pregnancy or first pregnancy late in life. No or short periods of breastfeeding. Or family history of breast cancer. %70 of women with breast cancer have no risk factors. 10% of them are due to hereditary defects. There are mutations in the genes BRCA1 and 2. Basically, 50 to 80% of women with these genes develop breast cancer. This also increases the risk of ovarian cancer. Now, how you diagnose breast cancer Early detection is self-examination, and that's why when you start getting women checks, they'll ask you if you're doing this, and if you don't know how to do it, they will show you. And mammograms, which is a type of examination. Now, the American Cancer Society recommends screening every year for women over 40. The U.S. Prevention Service Task Force on Breast Cancer Screening recommends delaying mammograms until age 50, but it's still the norm to do them at age 40. The diagnostic MRI recommended, it's only recommended for women of high risk factors. And so here on the right, you can see the mammogram procedure. It is not a friendly, happy procedure to do. They essentially put the breast between two plates and squish it and try to get as much in there to squish it. And then you have to stand there for a few moments while they scan it. And you can see on the left on B that they're showing you a film of a normal breast, and then you can see the malignancy there in the film of the breast with the actual tumor. And your treatment depends on the characteristics of the lesion. Basically, until the 1970s, standard treatment was radical mastectomies that removed the breast with all underlying muscles, fascia, and associated lymph nodes. There's also a lumpectomy that's less invasive and just takes out the cancerous lump. Let me go back in here. This. And I'm going to put LN for lymph nodes. So that's what they did till the 70s. Now, when they're looking at characteristics of lesions, right, how they treat it, they can treat with radiation, chemotherapy, or surgery. It's often followed by, if you do surgery, radiation, and chemotherapy to try to destroy any stray cells. There are drugs for estrogen responsive cancers. So you have Herceptin, that's for aggressive cancer cells. Tamoxifen, which improves outcome for premenopausal women with early or late stage cancer, or Femara, which reduces occurrences. And like I said, a common surgery that they did until the 70s was the radical mastectomy. Lumpectomies basically is less invasive, and it only takes the cancerous lump. And simple mastectomies basically remove breast tissue and auxiliary lymph nodes. And a lot of times, or sometimes, women opt for breast reconstruction after this to help them feel more comfortable going back into society after all of this is over. Of this lecture for female reproduction. Now, it's always been assumed that females' total supply of eggs was determined at birth. And just to let you know, there is some new evidence that suggests stem cells can arise from epithelial cells on the ovary surface. So this may in long term overturn previous assumptions. Now, the whole point of female physiology is what's known as oogenesis. And this is a production of female gametes that takes years to complete. So this is basically the counterpart to spermatogenesis, but this is female. It begins in fetal period with oogonia. And this is a 2N diploid ovarian stem cell that is multiplied by mitosis and stores nutrients. Now, we will get from there, once mitosis happens, the primary oocyte that will then develop into a primordial follicle that becomes surrounded by follicle cells. Remember, we talked about that. And the primary oocyte begins meiosis, but stalls in prophase 1. At birth, females presumed to have lifetime supply of primary oocytes. Now, each month after puberty starts, a few primary oocytes are activated. One from this group is selected each month to become a dominant follicle that will resume meiosis and end up on its way to become a mature ovum. Now, after division of meiosis 1 is complete, 2 haploid cells of different sizes are produced. One is called a secondary oocyte, which is a large cell with almost all of the mother cell's cytoplasm and organelle, and the other is called the first polar body. And this is a small cell, almost devoid of cytoplasm, and it will not end up being a functional ovum in the end. After puberty, the secondary oocyte arrests in metaphase 2 and then becomes an ovulated ovum. If not penetrated by a sperm, it deteriorates. But if it's penetrated by a sperm, the second oocyte completes meiosis 2, yielding a functional gamete or an ovum, and a second polar body, then again, that is not functional. So through all this division, in sperm, you would end up having 4 functional sperm at the end, right? In female, you're just going to, after all this division, get 1 functional ovum. So that can also explain why the sperm count is so high, where in females, there's a lot of energy that's devoted to just making 1 cell, and that's why we females have limited number of gametes. And so these are the events basically for oogenesis so that you can see them, right? And so essentially, if you look over here, before birth, you have that oogonium. That's the stem cell, right? And then basically, it's going to perform mitosis and make a primary oocyte that is still diploid. Now, at this point, the primary oocyte that you can see, do you see, and this is kind of lining it up so that you can see what's happening inside the cell versus what the follicle development is in the ovary. So at this point, at the primary oocyte, you can see the oocyte and the follicle cells being around there, and that's the primordial follicle that's holding the primary oocyte. Now, in infancy and childhood, ovary functionally are inactive. So you can see that the primary oocyte now is arrested in prophase 1, and this is present at birth, and that these follicles are still primordial follicles. Remember, that's when the follicle cells, essentially, there's just 1 line of them around holding the oocyte. Now, early month from puberty to menopause, what happens? Well, at this point, the primary oocytes are still arrested in prophase 1. So you see here, they're still at that primary follicle stage. Now, at this point, basically, what's going to happen is that you can see over here, the follicle is now becoming the secondary follicle, which is going to have essentially those granulosa cells surrounding it versus the regular follicle cells, right? And basically, this is going to become what's called a vesicular follicle, which has this extra area. This is also known as an antral. And basically, at this point, they are making that antrum, which is that kind of fluid area to help them. And so you see at this point, we're still at the 2N stage, where we're still at a primary. Now, meiosis 1 happens at this point when luteinizing hormone surges. And so at that point, you're going to get a division. And then this side is going to be the end, but it's going to be a polar body. That's inactive. It doesn't do anything. And then you're going to get this basically secondary oocyte. And that's going to be the large cell that is mostly all the mother's cell cytoplasm and organelle. And you can see that it's really big. And at this point, there's going to be a dominant follicle that is basically going to be picked, right, for meiosis 1 to happen. So basically, this guy, the secondary oocyte, is going to rest in metaphase 2. And this is only going to become the ovulated, it's going to become the ovulated ovum. And so you can see ovulation happens here at the secondary oocyte. And you can see that it's coming out of this basically follicle that's here. And so again, that other polar body goes, and these guys are going to become, they're going to degenerate. They're not useful for anything. And then you're going to have meiosis 2 completed only if a sperm penetrates the oocyte. And then you're going to get the secondary polar body that doesn't do anything, and then this ovum. Now, once this comes out, you can see it's left. The corpus luteum that's there, that's basically this ruptured follicle becomes that, and essentially will degenerate, go away. So that's the stages of oogenesis. Now let's compare oogenesis to spermatogenesis a little. The number of functional gametes do different. Oogenesis produces only one viable ovum. You saw in 3 polar bodies. The polar bodies degenerate. They don't do anything. So we waste, or females waste, a lot of energy just to produce one egg, which is also the reason why women tend to be more selective when they go to copulate and when they go to actually have a child. There's unequal division will ensure oocytes have ample nutrients for 6 to 7 day journey to the uterus. It's a long move from the ovaries all the way to the uterus, right? Polar bodies degenerate and die, they're gone. Spermatogenesis produces 4 viable sperm. So in that case, they're not using a lot of energy for not a lot of yield. So they get it. Lots and lots of sperm made this way. Error rates differ. Spermatogenesis has an error rate of only 3 to 4%. So you're not going to see as many genetic abnormalities happen and or it not completing correctly. Oogenesis has an error rate of about 20%. But when you look at it, it is a lot more involved than spermatogenesis is. Now, the ovarian cycle is the monthly 28-day series of events associated with that maturation of the egg. There's 2 consecutive phases with ovulation occurring in the middle of the 2. There's what's known as the follicular phase. That's the period of vesicular follicle growth. That's day 1 through 14. And then luteal phase, which is the period that the corpus luteum is active. And that's 14 through day 28. Now, just to let you know, we do this average of 28 days, but only about 10 to 15% of women actually have this 28-day cycle. The follicular phase varies, but the luteal phase is always 14 days from ovulation to the end of the cycle. And so the follicle phase of ovulation cycle kind of looks like this. And we have the comparison up here. So let's talk about it really quick. So if you look at this follicular phase right here, we start up here with our primary follicle, right? And so the primary follicle comes from the primordial follicle. And we're going to look at this again here in a second, basically. But what happens here is that basically you have this really smooth kind of cells that surround it, and they're going to change shape, right? And basically from here, this takes about a year, and the primary follicle will then go to the secondary follicle. And so the secondary follicle is now there's more than one layer around it. So this is when the follicular cells will turn into granulosa cells here. And basically at this time, the granulosa cells and the oocytes are going to guide one another to help them basically develop. From here, the secondary follicles become the vesicular follicle. And these early vesicular follicles form when clear fluid starts to accumulate. And that's basically forming what's known here as an antrum. And this is kind of where we start their antral phase, which is completely regulated and is dependent on hormones. Primary and secondary follicles are not at all dependent on hormones. So let me write this. On hormones. At this point, basically that antrum is going to have this large cavity that forms. And this is what helps you to distinguish this vesicular follicle from other types of follicles in the past. And basically what's going to happen at this point is ovulation is going to occur. And this is when we start the luteal phase. And so you can see here that it turns into the corpus luteum. And basically, this is going to, it kind of becomes a temporary endocrine gland because it can release hormones. Now, if there is no pregnancy within about 10 days, it starts to degenerate. And so this kind of shows you all the different stages between those. Now let's look and get just a little bit more detailed. You knew that was going to happen, right? And so here we're going to take a gander at what we were just talking about and look at it in terms. And you can find this on your book on basically 1057 if you're using the Human Anatomy and Physiology 10th edition of Mirab. This is Pearson. And so if you look at number 1 here, you'll see that is those primordial follicles. And basically, once you have those, if you look, they are going to go into step 2, which is the primary follicle, or the primordial follicle becomes a primary follicle. So essentially, and it's showing, not really showing you, it starts here at number 2. So if you look at that first picture here, you can see that around the oocyte, you have these squamous-like cells that are very flat. And then when you go to the primary, they become very much cuboidal, and that oocyte starts to enlarge. This process takes about 1 year. Now, the primary follicle then can become the secondary follicle. And you can see the secondary follicle right here. And it's included right there. And so at this point, the follicle cells are going to start multiplying, proliferating, getting more. And so they're going to form here stratified epithelial. And so this is when you go, remember, if you have one layer, it's follicular cells. But at this point, do we have one layer? No, it's stratified. There's several layers there. So at this point, this is when we're going to see the granulosa cells we were talking about. Now, the granulosa cells and the oocyte together guide one another's development, and they guide it through what's called a gap junction. Okay, and so at this point, the secondary follicle becomes a vesicular follicle. And you can tell this because you start seeing certain things forming in here. So let's look at this picture a little bit. So basically, you're going to have connective tissue, and granulosa cells are going to start condensing. And that's what forms this area right here. And this is what's called a theca folliculi. So connective tissue plus granulosa cells condense. Now, around the O site, which you can't really see, but yeah, you can, it's right there, is what's known as a zona pellucida. And this is just kind of this thick, it's a glycoprotein rich, it's kind of viscous membrane secreted by the oocyte, and it helps to encapsulate it. And so you can tell what happens here is these early vesicular starts forming this antrum. And like I told you, this is a follicle formed when clear fluid starts to accumulate. And that's how you can tell. That's an indication that it's a vesicular follicle. And so the antrum is going to continue to expand with fluid, basically isolating the ovum. And you can see that here in 4B. Now, on one side of the ovum, you have what's called a corona radiata. And basically, this is kind of sits on the stalk and one side of the follicle. Now, when the follicle is a size that's greater than about 1 inch or 2.5 centimeters, it bulges. And so that's when you'll start seeing this happen. And that's when it's ready to ovulate. You write this, bulge, ready to ovulate. Now, during this follicular phase, there are several of these follicular follicles that are stimulated to grow. And basically, they're triggered by follicle stimulating hormone. Now, these levels fall around middle of this follicular phase, and that's going to cause, so let me go, in the middle, you get a drop. And that's what helps decide the dominant follicle to be selected. How it truly works, they're still uncertain. And then from there, the primary oocyte of the dominant follicle completes meiosis 1 and 2 to form the secondary oocyte and the polar body. And basically, there are going to be granulosa cells in here that signal to the oocyte to stop in metaphase 2. Remember, we talked about that earlier. At this point, you're going to have ovulation. And ovulation occurs due to a rise in luteinizing hormone. Think, this first part is the follicular phase, right? And think follicle stimulating hormone is going to be in here. That's going to help develop the follicle. Well, if we release the ovum, we're going to have the corpus luteum. So think the luteus phase or luteinizing hormone. It all kinds of goes together there. And so those rising levels are going to cause the ovary walls to rupture, expelling the secondary oocyte here. And you're going to see here, that's why I mentioned it, the cornea why can I not say that right now? I think it's because it's so late at night. Radiata is going to release. Now, there is this weird phenomenon that happens. It's called Mittelschmerz, and this is basically the tinge of pain that happens to some women during ovulation, like they can feel it happening. I've had it before, so it's not completely unheard of. Now, about 1 to 2% of ovulation can release more than one oocyte, which if fertilized can result in fraternal twins. Now, identical twins result from fertilization of one oocyte, then separates daughter cells, right? So that can tell you the difference between having fraternal twins, right? So that's 2 ovums released, and this is 1 to 2%, right? Or identical twins is 1 ovum, right? And it ends up releasing later. So in this luteal phase, after ovulation ruptures and the follicle collapses, the antrium will fill with clotted blood. And you can see where ovulation is happening here. Now, this is known as corpus is known as hemorrhagicum. But this essentially will be absorbed. Now, the remaining granulosa cells and these thecal cells enlarge to become the corpus luteum. And basically, this is important because it will secrete progesterone and some estrogen. Now, if there's no pregnancy, then it disintegrates. And basically, that's what's known later as and I'm just going to write it here on the front of it, corpus albicans. And this is a scar, and this happens in about 10 days. And then this luteolytic or ischemic phase, that's about 2 or 3 days of the luteal phase, is when the endometrium actually begins to erode. And just to let you know, if pregnancy does happen, the corpus luteum begins producing hormones that help sustain pregnancy until the placenta takes over. And that's about 3 months. So it almost like it becomes a temporary endocrine cell there. That was a lot, I know. Now, how do we regulate that ovarian cycle? I did talk about a few hormones in there. So, before puberty, ovaries secrete small amounts of estrogen. And so basically that helps to inhibit the release of the gonadotropin release hormone from the hypothalamus. As puberty nears, if leptin levels are adequate, the hypothalamus becomes less estrogen sensitive, so that gonadotropin releasing hormone is released. This will stimulate the follicle stimulating hormone and luteinizing hormone that's released by the pituitary that acts on the ovaries. Do you guys realize that these are the same hormones we talked about earlier, right? So the events occur until an adult cyclic pattern is achieved. Or and menarche occurs. So let's talk about how we regulate the ovarian cycle itself. So if we're in this early or mid follicular phase, this is what's happening, guys. The hypothalamus is talking and releasing the gonadotropin releasing hormone. It travels via the portal blood to the anterior pituitary system, where it's going to release follicle stimulating hormone and luteinizing hormone. That's going to help stimulate follicles to grow, mature, and secrete sex hormones. If we go over here to the follicle stimulating hormone, what this does is it's going to help basically stimulate the granulosa cells to release estrogen. Now, on the other side, you have the luteinizing hormone over here, and they are going to prod the thecal cells. And the thecal cells are going to release androgens, which the granulosa cells are going to convert to estrogen. Now, what happens here is when estrogen is released, it's going to create a negative feedback loop. And basically what it's going to do, if you see it goes up here, and basically it's going to inhibit the release of follicle stimulating hormone and luteinizing hormone. Now, the inhibin gets released from granulosa cells, and that just inhibits follicle stimulating hormone. Now, this can stop basically all the developing follicles to basically all the, let me write it up here. At this point, when this happens, the developing follicles will start to deteriorate, except for the dominant follicle. The dominant follicle can withstand the FHS dip. Now, once we get into the late follicular and the luteinizing phase, what happens here is that you are going to get release now of the gonadotropin releasing hormone. And what this is going to do is basically estrogen levels at this point are going to continue to go up, which the mature or dominant follicle will keep releasing estrogen levels. And when they get to a critical high value, a positive feedback will happen. And so this is going to allow the gonadotropin hormone to release, and basically you're going to get a surge of luteinizing hormone. And this is going to trigger ovulation. And formation of the corpus luteum. So at this point, right, this luteinizing hormone surge is going to trigger the primary oocytes to complete meiosis 1, to become secondary. Secondary, right? And then the secondary oocytes will enter into meiosis 2 and continue on to metaphase 2 of meiosis 2. And so there's a lot of other things that happen, but we're not really going to talk about this because I feel like there's already enough going on here to basically talk about this ovarian cycle. So the rest of the things you can go and look to kind of see the other things that this luteinizing hormone does, but we're going to stop with that. We're just going to talk a little bit about the formation of the corpus luteum at this point. So shortly after ovulation happens, you have this corpus luteum that forms. Now, the luteinizing hormone stimulates the corpus luteum to secrete progesterone. And the progesterone is going to help maintain the stratum functionalis. And remember, the functionalis was the area that's inside that gets shedded whenever a woman ends up having her menstrual cycle, or the ovum embeds into whenever it's fertilized. And so to maintain pregnancy, if it occurs, negative feedback inhibits luteinizing hormone and FISH is released. So let me put up here. So if pregnant, right, you'll get inhibit of LH and increase of FSH. And basically this inhibin here will enhance the inhibitory effects so that the pregnancy can continue. Basically what's going to happen is declining luteinizing hormone is going to end the luteal activity and inhibits follicle development. So basically, as this plasma progesterone continues, estrogen levels will help to inhibit You can see that in the little line there. So if no fertilization happens, this guy goes away, right, when LH levels fall. And basically what happens is because you're not making progesterone and estrogen, essentially the FSH and LC secretion, that blockage stops and it causes the cycle to start all over again. Oocytes are actually activated 12 months prior to ovulation, but mature 14 days into ovulation. And so that's essentially how we regulate. I told you the women were more complicated here. And so this kind of gives you an idea of what happens. So, during that follicle phase, you can see they're kind of lower. Here is in the middle is ovulation, and then it goes back down again. Whew. Now let's just talk about the menstrual cycle, and it's basically the cyclic series of changes in the endometrium, and it occurs in response to the fluctuating ovarian hormones that we're talking about. And so there's 3 phases to the menstrual cycle if there's not a pregnancy, right? Days 1 through 5 are menstrual phase. Days 6 through 14 are the proliferatory or ovulatory phase. And day 15 through 28 is the secretory or post-ovulatory phases. So we're going to talk about each one of these. And so this is how thick the functional layer and the basal layer of the endometrium is. And so when we look at this, you can see day 1 through 5 right here. This is when the ovarian hormones are lowest. This is when the gonadotropin levels begin to rise. And basically the stratum functionalis detaches from the uterine wall. See, and it says it right there. And so basically menstrual flow of blood will last from day 3 to 5. By day 5, the ovarian follicles start to produce more estrogen. So the rise of estrogen is going to allow for this new generation of the stratum functionalis layer to basically thicken. The glands are going to start enlarging. If you can see right here, you can see that the spiral arteries are going to start increasing in number. Now there's normally kind of this thick, sticky cervical mucus, and during this time, it's going to thin it out to help facilitate sperm movement in here. And ovulation is going to happen at the end of the proliferous phase. So secretory phase begins immediately after ovulation, and this is day 15 through 28. This is the phase that's most consistent between women. Basically, what's happening is the endometrium is preparing for an embryo to be implanted. And so during this phase, you're going to see rising progesterone levels from the corpus luteum. And that makes this functional layer here kind of start becoming a secretory mucosa. And it's going to start allowing these glands to become bigger and secrete nutrients. Basically, all of this is happening just to make, and this is the most important part of that, basically what's happening is that thickened mucus helps to form a cervical plug that blocks sperm entry. We don't need any more sperm. We don't need any pathogens or any debris. So it's going to block it all during that time. During this time, and so if fertilization doesn't occur, then instead of the corpus luteum helping with all of that, it's going to basically degrade at the end, and progesterone levels fall again. And basically, this is going to allow those spiral arteries to start kinking and spasming. And then from there, the endometrial cells die, and the glands recess, and then they'll relax, and then the whole thing like sludges off. And the whole thing starts over again. So this is basically the 3 different main stages and what happens during them. And this is just there to help you if you want to kind of look at everything happening all at the same time to relate the fluctuations of the gonadotropin levels with the luteinizing hormone in fish between the follicular stage and the luteinizing phase, looking at how the follicle changes, and then the plasma hormone levels of progesterone and estrogen, and basically looking at how that affects the formation or what happens during the luteinizing phase, and then looking at how the different phases during menstruation are affected during these different stages. Now, extremely strenuous physical activity can delay menstruation in girls and disrupt normal menstrual cycles in adult women, or miniarchy in girls and disrupt normal menstruation cycles in women. And it can cause amenorrhea, which is the sensation or the stop of your menstruation or menstrual cycle. It can also cause a problem. Female athletes tend to have lower body fat levels, and basically adipose cells are needed to convert adrenal anagen to estrogen. Also, leptin plays a role in female puberty. It helps to inform the hypothalamus if energy stores are significant to support reproduction. If they're not, the reproductive cycle is shut down. Also, this can lead to osteoporosis. Now, estrogen is important because it promotes oogenesis and follicle growth in ovaries. It helps to increase anabolic effect on female reproductive tract, and it's there to support rapid, short-lived growth spurts of puberty. It also helps to induce secondary sexual characteristics like growing a breast, having subcutaneous fat on your hips and breasts, and the widening and lengthening of the pelvis. And we looked at some secondary sexual characteristics in male as well. There's also metabolic effects. It helps to maintain low blood cholesterol levels and high HDL. Remember, those are good to help get rid of cholesterol. And it facilitates calcium uptake. Progesterone is important because it works with estrogen to help establish and regulate uterine cycles. So it helps to promote the changes in that mucus, essentially the mucus plug that we were talking about. And it also has effects on placental progesterone during pregnancy. It can inhibit the motility of the uterine and helps prepare breasts for lactation. Now, the female sexual response is limited by both psychological stimuli and touch. So anything, the clitoris, the vaginal mucosa, bulbs of vestibule, and breast engorge with blood during the sexual response, and nipples become erect. The vestibular gland secretes the lubrication to help lubricate the vestibule. And orgasm is accompanied by muscle tension, increase in pulse rate and blood pressure, and rhythmic contractions of the uterus. Females are lucky. They have no refractory period after orgasm and can experience multiple orgasms in a single sexual experience. But orgasm is not essential for conception, where it is for males. The female libido is prompted by DHEA. It's an androgen that's produced by the adrenal cortex. And so we're going to go really quick over some STIs, and these are sexually transmitted diseases, is another name, or venereal diseases. U.S. has the highest rate of infection among developed countries. Latex condoms can help prevent spread. STDs, or Is, are the single most important cause of reproductive disorders. You have gonorrhea. One second here. It is a bacterial infection of the mucosa of reproductive and urinary tract. It's caused by Neisseria gonorrhoeae, and it's commonly called the clap. It's spread by contact with genital area or pharyngeal mucosa, and it's treated by antibiotics, but some strains are resistant. They're becoming more prevalent. Number of cases in the U.S. have been declining because of the availability of antibiotics. Some of the signs and symptoms for male: urethritis, painful urination, or discharge of pus. For females, the majority of females display no signs or symptoms. They might have some abdominal discomfort, vaginal discharge, or abnormal uterine bleeding. The bad thing, it can result in pelvic inflammatory disease and sterility. Syphilis is a bacterial infection transmitted sexually or congenitally. It's caused by Streptomonas pallidum, and it can infect fetus. Infected fetus can be stillborn or die shortly after birth if they're infected with syphilis. The bacteria invades the mucosa or even broken skin. Infection is asymptomatic, which means you have no symptoms for 2 to 3 weeks, and then painless transfers appear on the site of infection, but they can disappear within a few days. If untreated, secondary signs appear several weeks later. So these are kind of like pink skin rash, fever, and joint pain can develop. It usually appears between 3 to 12 weeks, then disappear. The disease can have a latent period, which may or may not progress to what's known as tertiary syphilis. It's characterized by gumas, which are lesions that develop in the central nervous system, blood vessels, bones, and skin. It's very much treatable through use of penicillin, but the number of cases have been increasing. The most common bacterial STI in the United States is chlamydia. It is caused by Chlamydia trachomatis. It can be contracted from birth canals by newborns. It's responsible for about 25 to 50% of all diagnosed cases of pelvic inflammatory disease or PID. Symptoms are urethritis, penile and vaginal discharge, abnormal rectal or testicle pain, painful intercourse, and irregular menses. 80% of women have no symptoms, though. Left untreated, it can lead to sterility. Now, this can also cause arthritis and urinary tract infections in men. Like I said, newborns can pick it up from the birth canal, and they can develop what's known as trichomonas. And this is a painful eye infection. It can lead to corneal scarring. You can also get respiratory tract inflammation, including pneumonia from this. And the treatment is tetracycline. Trichomoniasis is the most common curable STI in active young women in the U.S. About 7.4 million new cases are diagnosed each year. It's a protozoan infection, so it's really easy and inexpensive to treat. Symptoms are yellow-green vaginal discharge with a really strong odor. However, some patients can be symptomless. And genital warts is caused by the human papillomavirus, HPV. It's a kind of a string of herpes virus. It's the second most common STD in the United States. About 6.2 million new cases each year are actually diagnosed. Realize because so many people are asymptomatic that a lot of people go undiagnosed. 80% of cases of invasive cervical cancer are linked to some strain of HPV. Most strains do not cause cancer. It's only a couple of them. One, it's like 16 and 18. One causes genital warts. One causes cervical cancer. And there's a couple more. I just can't think of them off the top of my head. Treatment, difficult and controversial. Some physicians prefer to leave the warts alone. Some prefer to do basically laser surgery or freeze them off, cryosurgery, or alpha interferon treatment. The cryosurgery and laser can be painful from what I hear. Genital herpes is caused by herpes simplex virus 2. Possibly 1/4 to half of American adults carry this virus. Only 15% display signs of infection. It's characterized by latent periods and flare up with vesicle formation. They can be passed to the venous, and basically congenital herpes can cause malformations. They can be treated with acyclovir and other antiviral drugs. And so the last part of this is talking about genetic sex and embryological and fetal events that happen. And so we, if you remember, have 46 chromosomes in fertilized eggs. 2 are sex chromosomes. The other 44 are autosomes. So you have the final sex chromosome. It can be, the X chromosome is large and the Y is quite small. So in females, it's XX. Each ovum always has an X chromosome. And for males, they're XY. So 50% of the sperm contains X chromosomes and 50% contain Y. Now, this determines the genetics of sex. So basically what it's saying is if you get an X from the egg and X from the sperm, you're going to be a female. If you get an X from your egg and a Y from the sperm, you're going to be male. And this SRY gene is a master switch on the Y chromosome that will initiate testy development and maleness. So the father determines the sex of the child. And in history, where all these women were decapitated because they could not produce a male heir, it was actually the king's fault. So there are some homeostatic imbalances that happen to this. It's called non-disjunction. And this is when during gamete formation that the meiosis doesn't happen correctly. There's actually 2 types. Non-disjunction type 1, which means during the first stage of meiosis, it doesn't separate outright. Non-disjunction type 2 is during the second stage. Non-disjunction type 1 actually makes it so that the majority of all the gametes are messed up, where in non-disjunction type 2, only 2 of them are. And it can lead to different syndromes. So most of the time during non-disjunction, it's going to lead to spontaneous abortions. But there are times where those actually survive. Turner syndrome, for instance, is female. And so in this case, they only get one single X chromosome. You need an X chromosome to survive. You do not need a Y to survive. And basically in females, whenever you have the XX, one of them is going to be inactivated. And that should make sense because in males, they only have one. Now, in some species, they end up basically, some will become inactive, others won't. And you can see that happening in something like a calico kitty cat. It's like the black ends up having one X that's activated and the orange part is the other one. So you can see where it's different. We don't have anything really like this. But in Turner syndrome, what happens is you only have one X, you don't have the other. So because of this, they won't ever end up developing ovaries later, so they will become sterile. So, females with 4 or more X chromosomes are intellectually disabled and have underdeveloped ovaries with limited fertility. Klinefelter's is male, and so basically this is when it's XXY. They're sterile and are normal or only slightly below normal intelligence. And a lot of times with them, you can tell males tend to have more like kind of feminine characteristics, as in their face is a little bit, instead of it being so rigid, it's a little bit more smooth. Their hips tend to be a little bit wider apart, kind of look more like hippie instead of being straight down forward. And so that's what they look like. And so basically during the development of the reproductive system, sexual differentiation of the reproductive system includes basically there's sexual in different stages where the embryo could develop into male or female. And so you have these paramesophrenic ducts, basically future female ducts, form lateral to mesophenic ducts for future males. So basically, there's going to be a point where the decision is made. And so Mullerian ducts are basically the areas that are going to be basically female, and Wolffian ducts are going to be males. And the gonads begin developing in week 5 as gonadal ridges form. Basically, the primordial germ cells will start migrating to the gonadal ridges to provide germ cell destined to become spermatogonia or oogonia, right? Testing formation begins in about week 7 for male, and female ovaries start to develop in week 8. And so if you have the SRY gene about week 5 to 6, here, there's indifferent, right? So you look here, you'll notice the Mullerian. This is future female ducts. The Wolffian, this is future male ducts. They're both there, right? Nothing looks different. Whether you have, now this is going to be what ends up being the male, and this is what's going to end up being the female, right? So about week 7 to 8 for males, you'll notice here that these particular ducts are starting to degenerate. And these are forming the vas deferens, where in females, these are starting to degenerate, and these are making the uterine tubes. So this is a differentiation that happens due to the presence of that SRY gene. And you can see the locations are different. For instance, the urinary bladder in males in the middle, it's starting to move over to the side of the where the uterus is starting to form with that mesophenic duct. All right. Now at birth, the difference between, you can see here, hugely different between the development of the two forms, right? So basically, the sexual differentiation, external genitalia start to form in about week 8. And so the genital tubercle becomes the penis of the male or the clitoris in the female. The urethral fold will become the spongy urethra of the male or the labia minora in the female. And the libido-scrotal swelling will become the scrotum in the male or the labia majora of the female. And so when we were talking about this one is like the counterpart to this one, kind of explains it. If testosterone is absent, all embryos develop into females because that gene helps to increase the level of testosterone. And so basically this just kind of shows you the development of the homologous structure of the external genitalia in both sexes. And you can see up to about week 5, they're the same, you can't tell. And then the activation of testosterone will happen. And then you can see basically the glans penis forms, and you can look over here and see all the different things that we've just talked about located in those different pictures. I'm not going to spend a lot of time on that. So there are homeostatic imbalances for this, pseudohermaphroditism, individuals with external genitalia that don't match their gonads, right? It's caused by interference in normal sex hormones produced in embryo. So embryonic testes do not produce testosterone. A genetic male develops female accessory structures and external genitalia. If a genetic female is exposed to testosterone, the embryo has ovaries but develops male ducts, glands, as well as a penis and an empty scrotum. And this is why many seek sex change operations to match outer self, the external genitalia, to their inner self or their gonads. At about 2 months before birth, testosterone will stimulate the migration of testes towards the scrotum. Migration is mechanically guided by fibrous cords called gubernaculum from each testis to scrotum. Ovaries also descend but stop by those broad ligaments. Cryptochidism is the failure of testes to make normal descent. It can cause sterility, increased risk of testicular cancer. Surgery can be performed. And basically, and we're almost to the end, guys, during puberty, like we talked, fish and luteinizing hormones are elevated at birth but drop and remain low during prepubescent years. Puberty is basically the period when the reproductive organs grow to adult size and become functional. And it occurs in response to rising levels of gonadal hormones, the follicle-stimulating hormones and the luteinizing hormones. Secondary sexual characteristics will appear during this time. This is the earliest time that reproduction is possible. And menopause for women is when menses has ceased for an entire year. It's equivalent to no menopause. There's no equivalent to menopause in men. And basically, there's a declining estrogen levels, and it can have a full body effect. It causes atrophy of reproductive organs in breast. It'll cause irritability and depression in some, hot flashes. And this is because as skin blood vessels undergoes intense vasodilation, it can feel like it's really hot. Gradual thinning of skin and bone loss, and increased total blood cholesterol levels and falling HDL. So it can cause problems with cholesterol.